Good day, students, and welcome to Clinical Bacteriology Week 2 lecture session. Now, in this lesson 2, we will be talking about the different aspects of the bacterial cell, from its structure and components, as well as its growth requirements and biochemical processes. Now, the knowledge of the bacterial cell is very important, especially in diagnostic microbiology, as it aids in microbial identification. Now, as we previously learned from lesson one, your microbes are quite ubiquitous. However, only a small portion of them are pathogens or capable of causing infections. Some microbes also can be found as normal inhabitants of the human body, as normal flora, and many can be found in the environment. However, again, in diagnostic microbiology, our only main focus will be those that affect human health. Now, to define, pathogenic bacteria are prokaryotic cells that infect eukaryotic hosts such as humans. Now, structurally, the bacteria and its host cell differs in that prokaryotic cells are much more simpler and lacks a membrane-bound nucleus. Now, human cells, however, are much more complex due to their eukaryotic nature. Now, this difference in structure is the reason why many antibiotics are effective in treating bacterial infections, as antibiotics targets processes or structures unique to the prokaryotic cell and therefore leaving the eukaryotic cells or host relatively unharmed. Now let us proceed with the structure of a bacterial cell. Now, this table summarizes the differences between the prokaryotic and eukaryotic cell organization. Now, in terms of size, we can see that the prokaryotes are much smaller, ranging only from zero point 0.4 or 0.20 to 2 micrometer in diameter. Now, eukaryotes, however, range from 10 to 100 micrometers in diameter. Now, the cell wall of your prokaryotes usually uh, contains the peptidoglycan structure, while your eukaryotes naman in terms of cell wall, most of your eukaryotes doesn't have cell wall, except for your fungi, which contains the chitin molecule. Now, if present po in your eukaryotes, it is usually chemically simpler. Now, the nucleus naman po is absent in prokaryotes, meaning that they do not have membrane-bound nucleus, and the genetic material of your prokaryotes usually is situated in a structure called nucleoli. Now, your eukaryotes naman typically, their genetic material is protected by a membrane, thus it contains membrane-bound nucleus. Now, for the genome po of your prokaryotes is located in the nucleoid, situated in parts of the cell membrane called mesosome. While your eukaryotes naman, its genome is located inside a membrane-bound nucleus. Now, chromosomal DNA of your prokaryotes is much simpler and circular with a complex RNA. And its simple and circular chromosomal DNA of your prokaryotes is usually doesn't have histones. So it is not organized by your histones. However, since the chromosomal DNA of your eukaryotes is more sophisticated and it is in terms of base pair, it's much longer. It needs histone for organization. Now, for your prokaryotes, generally, they do not have membrane-bound organelles unlike your eukaryotes. Now, in terms of ribosomes, the ribosomes of your prokaryotes is much smaller compared to the larger eukaryotes. Now, in fact, the ribosomes of your prokaryotes is in terms of size, it is 70 Svedberg units, while your eukaryotes naman is 80 Svedberg units. Now, the lysosomes and peroxisomes is absent in prokaryotes. That's why it doesn't have um, protective organelles to protect its structure and also its function. While in your eukaryotes, no, it is present naman. Now, some of the special appendages or parts of the prokaryotes are your pili and fimbriae, which is present almost some of your bacteria or your prokaryotes has pili and fimbriae. Now, the function of these parts of the prokaryotes will be mentioned later. Now, in terms of having a part or a part for locomotion, both prokaryotes naman po and eukaryotes have flagella. Okay? But in terms of complexity, your eukaryotic flagella is much more complex, containing multiple microtubules, where your flagella of your prokaryotes naman is much more simpler, consisting of two protein building blocks. Now, in terms of the presence of sterols on cell membrane, now most of your prokaryotes doesn't have sterols on its cell membrane, except for the family of Mycoplasmataceae, which contains mainly sterols on its cell membrane. While your eukaryotic cells naman po contains a lot of sterols of its on its cell membrane. Now, in terms of gene structure, some of the genes of your eukaryotes po contains introns, which are sequence of base pairs in DNA that interrupts the continuity of genetic information. Now, these introns po in the part of your genetic material of your eukaryotes is not present naman po in prokaryotes. So, meaning it doesn't have introns, no, to begin with. No, kapag ang isang genetic material po has introns, um, mas stricter po yung pag-check ng mga specific information in the genetic material. While in the prokaryotes, they do not have introns. So medyo sensitive po yung kanilang genetic material for in cases of mutations. Now, in terms of cytoskeleton and cytoplasmic staining, so usually po cytoskeleton is present in eukaryotes while absent in prokaryotes. Now, the presence naman po of gas vesicles no, is usually seen no, or typically seen in prokaryotes. Now, in terms of cell division, your eukaryotes po usually is complex in terms of cell division. It follows asexual or sexual reproduction. While your prokaryotes naman po is much simpler, it usually undergoes asexual division or reproduction through the binary fission process. Now, this is a diagram po showing the difference between prokaryotic bacterial cells and a generalized animal cell. You may go back to this diagram as I move on with the discussion so you will have a visual reference. Now, let's now first talk about cytoplasmic structures in a prokaryotic cell. Now, before we begin again, your prokaryotic cell is much smaller and less compartmentalized compared to eukaryotic cell, and they do not contain membrane-bound organelles and nucleus. Now, the first cytoplasmic structure inside your prokaryotes or your bacteria now is your ribosome. So, your bacteria po, meron pa rin po silang ribosomes which are sites of protein synthesis. Now, the ribosome sizes are usually expressed using the S unit, which stands for Svedberg unit. Now, this unit refers to the sedimentation rate of a particle during high-speed centrifugation. Now, the larger the particle size, the higher the S value. Now, for bacteria, the ribosome size is about 70S and is composed of two subunits. So, a larger subunit with 50S and a smaller subunit which is 30S. It is important to note, however, that S value are not additive due to the loss of surface area when two subunits combine. That's why 50S together with the 30S, the total unit or size of the ribosome of your bacteria is only 70S. Okay? And usually po, your ribosomes po gives the granular appearance in your bacteria when seen microscopically. Now, the second main structure in the cytoplasm of your prokaryotes or bacteria is their genome. Okay? Now, the chromosome usually po of your bacteria is circular and can be found in an area of the cell called the nucleoid. So, the nucleoid naman is situated in mesosome, which are organelles of your bacteria that appears as the invagination of the plasma membrane and functions either in DNA replication and cell division or excretion of exozymes. Now, starting from here, I'll talk about the bacterial genetic elements and alterations. Now, in this portion of the lecture, we will discuss now the terms and mechanisms involving the bacterial genetics. Now, in terms of the discussion about the bacterial genetics, there are three aspects. First is its structure and organization of that genetic material. Now, typically, the genetic material pa rin naman of your bacteria is composed of bases, your adenine, guanine, cytosine, and thymine, which are covalently linked to each deoxyribose sugar, which are the key to the genetic code. Now, a DNA sequence that encodes for a specific product is defined as a gene. Okay, to review lang po. Now, all genes taken together within an organism comprise a genome naman. Now, the size of a gene and an entire genome is usually expressed in the number of base pairs or the BP present. The longer the base pairs, the higher the number of the genome present. Okay? Now, the genome is organized in discrete elements known as chromosomes. And in terms of bacterial genetic material, now the chromosome of the bacteria is usually a single unpaired or haploid chromosome. Now, the bacterial chromosome contains all genes essential for viability and exists as a double-stranded, closed circular naked macromolecule, which is situated nga po in a nucleoid. Now, information of protein synthesis is encoded in the bacterial DNA and transmitted in the chromosome to each generation. Now, the general flow of information in a bacterial cell is from DNA to messenger RNA. Then the messenger RNA acts as the blueprint for protein construction, and this messenger RNA will be taken by your ribosomes for protein synthesis. Now, the second aspect of the study will be the replication and expression of the genetic information. Now, to define, no, replication is the duplication of chromosomal DNA for insertion into a daughter cell. So, this replication results from a division of one parent cell, and each daughter cell receives the full and identical genetic complement contained in the original parent cell. Now, the process takes approximately in your bacteria for a rapidly growing bacteria only in 40 minutes po. Okay? So the process of encoding information in genetic elements is known as gene expression. Now, there are two main phases of replication, which is your transcription and translation. So transcription po is the synthesis of single-stranded RNA using one strand of the DNA as the template. Now, the main goal of transcription is to convert the DNA-based sequence of a gene into a messenger RNA molecule. Now, an enzyme called RNA polymerase is the enzyme central to the transcription process. Now, the RNA molecule that is being copied in the transcription process will be later undergoing the second phase, which is translation naman. Now, translation naman po is the actual synthesis of a specific protein from the mRNA. The genetic code with mRNA molecule is translated into specific amino acid sequence, no? That is responsible for protein structure and function naman. Now, let's talk about naman po the different mechanisms by which the genetic information of your bacteria is changed or exchanged among different types of bacteria. Okay? So, first, let's talk about mutation po. So, mutation po is a change in the original nucleotide sequence of a gene or genes within an organism's genome or genotype. Okay? Now, the mutation po may be the result of insertions or deletions in the genome that leads to a disruption of the gene or a frameshift mutation or both. Now, it may be induced by chemical or physical factors in the environment or by biological factors such as the introduction of foreign DNA into the cell. Okay? So typically, no, your bacteria po undergoes spontaneous mutations, no, every now and then, occurring in bacteria at a rate of 1 in 109 cells. Okay? Now, it occurs as the result again of error during DNA replication. So, in the normal replication process of your bacteria, no, typically nagkakaroon po talaga ng errors. So, in a rate of about 1 in 107 cells. So, again, exposure to certain chemical and physical agents can greatly increase the mutation rate in your bacteria. Okay? So, let's talk about naman the recombination or what we known as homologous recombination. Okay? So recombination naman po is a method by which the genes po are transferred or exchanged between homologous regions on two DNA molecules. So this recombination po provides a way, no, for organisms to obtain new copies or new combinations of biochemical pathways which changes, no, in their environment. So upon the adjustment of your bacteria and evolution, no, some recombination no, happens to acquire new biochemical pathways for the bacteria to better survive in its environment. So it occurs when a portion po of the genetic material that originated from one bacterial cell or the donor's bacterial cell is transferred into a second bacterial cell or the recipient. And the recombination po involves a number of binding proteins with the Rec-A protein playing a central role in recombination. Now, this shows you a simple diagram on how your recombination happens. So, again, recombination needs two copies of each chromosome in your cell or in the cell of the bacteria. And then these chromosomes are known as homologous chromosome. So, during the genetic recombination, so nagkakaroon po ng physical exchange na of the pieces of genetic information to the two different copies of your chromosome. And then, on that exchange, nagkakaroon po ng genetic material transfer. Now let's talk about the three mechanisms of gene transfer in your that happens no typically in your bacteria. So the three mechanism of gene transfer is your transformation, transduction, and conjugation. Let's talk about it one by one. Now first is your transformation. So paano po ba nagkakaroon ng genes transfer through transformation? So transformation po, it involves recipient cell uptake of free DNA which are released po into the environment when another bacterial cell or the donor dies and undergoes lysis. Okay? So commonly po na mga bacteria that undergoes transformation to transfer its genes to others is your Streptococcus pneumoniae, Neisseria gonorrhoeae, and your Haemophilus influenzae. So these bacteria is capable of being recipients na of free DNA naman po. So transduction naman po is the transfer of bacterial genes by a bacteriophage from one cell to another. So transduction naman po, it involves na po a type of virus that specifically infect your bacteria, which is called bacteriophage. Now, in this mechanism naman po, DNA from two bacteria may come together in one cell. Now, there are two different types of transduction, which is your generalized transduction and specialized transduction. Now, generalized transduction po is the transfer of genes no by the normal uh, lytic phase of the virus po. So it happens during the lytic phase of the virus per se. So the bacterial DNA may be randomly incorporated with the viral DNA, and then usually po the generalized transduction ang nangyayari po diyan is that the bacteriophage will insert its own viral DNA, and then it will control no the bacteria by creating the necessary structures for it to replicate. So on that replication po, the bacteria po will now get the bacterial DNA or the fragment of the bacterial DNA, and upon doing its parasitic life cycle again to another bacteria, it will now enter or re-inoculate or re-enter the bacterial cell previously infected. Now, in specialized transduction naman po, usually it happens naman during the lysogenic cycle of the virus. No, ang nangyayari naman po dito is that the bacterial DNA will be inserted the bacterial or the viral DNA will be inserted adjacent to the bacterial DNA. So ang nangyayari po dito in the natural binary fission of your bacteria, nare-replicate din po yung viral DNA together. And then upon exposure to UV rays or UV or external or environmental factors, the viral DNA which is originally inserted po in the bacterial DNA will be spliced again, and then it will now undergo the lytic phase again. Now, this slide shows you the transformation summary. So in the left portion of the diagram, we can see the free naked DNA that is released by a lysed donor bacterial cell. Now, to ano lang po, to understand, again, for transformation, kailangan po nung donor cell na mamatay or malyse, no? Before it releases its naked DNA, okay, in the environment. And then when a competent bacteria is on your environment, now for a competent bacteria po, minsan po 'no, nakukuha po nila or naa-absorb po nila itong mga free floating na DNA na ito, and incorporating it to its own genome by the process of recombination. Now, the cell now has new genes that may alter its phenotypic characteristics, virulence, as well as its antimicrobial susceptibility. Now, this diagram naman po shows you the lytic and lysogenic pathways or cycles undergone by bacteriophages. Now, if you can see no, in the lytic cycle, no, the phages attach to and incorporate their DNA into the bacterial cell, and this DNA then directs the host machineries po to produce more phage DNA and protein coat, which will then be reassembled into new phage particles. Now, these new phage particles will lyse the bacterial cell, releasing the particles that are now ready to infect another host. And then when infecting another host, it will inject its DNA, and then the cycle continues. Okay? So in that mechanism, no, the bacteriophage will can transfer no the bacterial DNA from the previously attacked bacteria. Now, however, in some cases also your bacteriophage can undergo lysogenic cycle. Okay? So ganun pa rin po, it will infect the host cell and it may proceed to an alternate pathway. Now, in this alternative pathway of doing the lysogeny, now the phage DNA becomes incorporated in the bacterial genome together with the bacterial genome and becomes replicated together with the bacterial DNA during cell division or called lysogeny. Now, when triggered by certain conditions, the phage DNA becomes activated and napuputol po siya or na-excise po siya from the bacterial genome. Now, pag na-excise po yon or natanggal po, it will then enter the lytic stage in which the newly assembled phage particles may carry the genes again of its host DNA and then undergoing recombination in the process. Now, the third mechanism of gene transfer is called conjugation process. Now, in the conjugation process po, it is the transfer of genetic material from a donor bacterial strain to a recipient strain, and it requires close contact between the cells. So, it occurs between two living cells, and it requires mobilization of the donor's chromosome. Okay, so literally, this conjugation process is like a sexual phase now of transferring your genetic material or the bacterial genetic material of your bacteria. Okay. Now, it requires or it needs no a factor or fertility factor po for a bacteria to do conjugation or the F factor. Okay. So, the donor cell usually po has the fertility factor. It will establish po a conjugative bridge, no? Which is called a conjugation pili or sex pili that serves as the conduit for DNA transfer from donor to recipient cell. Now, classic example of genetic material transferred via conjugation are plasmids, okay? Which, when done, no, this is called horizontal transfer. So, but not all plasmids, however, are capable of conjugative transfer. Only those that have F factor na mga plasmids natin or F positive plasmids. Okay? So notes to remember, bacteria have used their capacity for disseminating genetic information to establish usually resistance to most of the commonly used antibiotics. So antibiotic therapy selects for bacterial strains containing plasmids encoding these antibiotic resistant genes. So this is one reason po antibiotics should not be overprescribed. Okay? So horizontal transfer of a plasmid is another way resistance to antibiotics is acquired through conjugation process, and usually both plasmids and chromosomal genes can be transferred by conjugation. Now, this diagram naman po shows you the transfer of DNA from donor to recipient strain. Okay? So, the material colored in blue is a plasmid that carries the F factor. Okay? Now, the donor strain produces a sex pilus or called the mating bridge, no, that enables the transfer of DNA also. So as we all know, both chromosomal and plasmid DNA can be transferred through this method. So as you can see in the diagram, the plasmid containing the F factor was transferred to the recipient strain. Okay, so after conjugation, both cells now have the F factor. So they are called F positive cells, no, and the capability to use conjugation as a means of gene transfer. Now, the donor are also called F positive cells or the male bacterium, and the recipient naman usually is called F negative or a female bacterium. Now, this shows naman po the summary, no, or an illustration of the three mechanism of gene transfer. So mainly namely the bacterial transformation process. So for transformation, it needs the donor cells to be lysed, or it needs to undergo lysis before it releases its DNA. For bacterial transduction naman, it uses viruses or the mechanism of bacteriophage to release po or to transfer genetic material from the donor cell to the recipient cell. Now, in terms of bacterial conjugation naman, it needs close contact and the production of the sex pilus or conjugation pilus to transfer genetic material, either plasmids or the main chromosome of the bacteria. Now, some terminologies connecting to bacterial genetics is your codon, no, anticodon, okay? Bacterial chromosome or the genome, the jumping genes. So, jumping genes are mobile pieces of DNA, no, that can literally jump from one place in the chromosome to another. So, most of your bacteria, no, contains jumping genes. So, one example of jumping genes present in your bacteria is called insertion sequence or IS. So, these IS po are 1,000 base pair mobile genes present in many types of bacteria, which can insert into a gene that can disrupt and inactivate the function of that gene. So, disruption and inactivation of the function of that gene so may cause the loss of the ability of that gene also. For example, loss of ability to ferment certain sugars. So another type of jumping gene is your transposons, which are located primarily in plasmids that often carry antibiotic resistant genes. So when we say point mutation naman po is a change in one nucleotide base that leads to a change in a single amino acid within a protein. So when we say competent cells naman, ito naman po yung kaya pong mag-take up ng mga naked DNA in terms of the transduction process kanina. Okay? So or yeah, it's the transduction process or transformation rather. Okay? So bacteriophage again is a virus that infects bacteria. And restriction enzymes naman po are enzymes that cut incoming foreign DNA at specific DNA sequence. And typically restriction enzymes are used to create used to create for insertion of new genes. Now, let's talk about now your plasmids. So, kanina pa natin po siya na-mention. So, what are plasmids po? So, plasmids po are extrachromosomal double-stranded element of DNA associated po typically with the virulence of your bacteria or your microbes. Okay? So, these are the sites for the genes for antibiotic resistance and toxin production. So, it is not essential for bacterial growth. Thus, it can be present or absent within the bacterial cell. And sometimes, it disappears during cell division and it can make bacteria mostly gram-negative pathogenic. So, it can make your bacteria or gram-negative bacteria pathogenic. Okay? So, it is a self-replicating and it can be transferred through vertical or horizontal transfer. Pag sinabing vertical po, it is transferred to daughter cell. So, upon the normal replication of your bacteria through binary fission, your plasmids can be transferred. Another way of transferring your plasmid is through horizontal transfer, which is mentioned earlier in the conjugation process. So, horizontal transfer is the asexual it is in the transferred of the plasmid through conjugation process. Now, there are two kinds of plasmids um, in terms of size. Now, meron po tayong large plasmid and small plasmid. Now, large plasmid typically is responsible to produce beta-lactamases, a type of enzyme that make your bacteria resistant to beta-lactam antibiotics such as your penicillin and oxacillin. Okay? Small plasmids naman po is usually dun po nakalocate yung ating mga resistance to tetracyclines and chloramphenicol. Now, another cytoplasmic structure po is your inclusion bodies or your cytoplasmic granules. Now, typically, inclusion bodies po are storage deposits po for energy source or food reserve of your bacteria. So, typically, it consists mainly of polysaccharides or glycogen and other lipids such as your polybeta hydroxybutyrate, or it can be made out of your polyphosphates or your volutin or metachromatic granules. Now, these polysaccharides po usually po are the another function of these polysaccharides or inclusion bodies is to lessen the osmotic pressure inside your bacterial cell. Okay? Now, these are the examples naman po of the different types of inclusion bodies that can be found no in some of your bacterial cytoplasm. So examples of inclusion bodies are glycogen inclusion bodies, cyanophycin granules, polybeta hydroxybutyrate granules, carboxysomes, gas vacuoles po, and also polyphosphate granules. Now, to mention lang po, some of these inclusion bodies can also be used to identify your bacteria. Kasi not all bacteria naman po has all of these inclusion bodies. Okay? So for example, your polyphosphate granules po have different compositions and appearance in certain types of bacteria. For example, in Corynebacterium diphtheriae, it contains or produces Babes urn granules, okay, or bodies. So another is in your Yersinia pestis na bacteria. So Yersinia pestis naman po produces primarily bipolar bodies, a different type of inclusion bodies or metachromatic granules. And lastly, one example also is your Mycobacterium tuberculosis. So it produces naman po a typical or a special type of inclusion body called Mutch granules. So your inclusion bodies mo can be used as an identifier for your bacteria. Now, another component of some bacterial cells is the endospore naman, a type of resistant structure or asexual spores. Now, these spores po are produced now by active bacterial cells as a form of backup. Okay? Now, when conditions become unfavorable to them. Now, if there is a lack of nutrient in the environment, or when the bacteria po are exposed to extreme temperatures, no, like lacking of moisture, or increased temperatures, no, the bacteria cell dies, but the spores persist due to their thick protein coat. Now, this highly resistant part of your bacteria, no, render them this highly resistant part of your bacteria render them resistant to chemical agents, temperature changes, starvation, as well as dehydration and UV and gamma radiation and desiccation. Okay? So, and then when favorable conditions are back, no, these spores po can germinate, and then it makes your bacteria again active, or it turn your bacteria into vegetative state again. Okay? So typically, endospores are produced by some of your gram-positive bacteria. Okay? And mainly it is composed of dipicolinic acid and calcium ions. Now, the size, shape, and interior locations of these structures can aid you, no, in the identification of some of your bacteria, like your Bacillus and Clostridium. Now, this slide shows you the development of some of your gram-positive bacteria in terms of sporulation. Okay, so the process or of spore formation is termed as sporogenesis or sporulation, while the end of the spore's dormant stage naman is called germination process. So as you can see here in this slide, no, your vegetative cell upon exposure to extreme environment or unfavorable condition, it will develop a spore undergoing the sporogenesis or sporulation process. Now, upon exposure of your spores into a favorable condition again, the mature spore will now undergo germination process, turning it into a vegetative cell again. Okay? Now, endospores can be classified according po to the position within the bacterial cell in which they can be found. Now, terminal spores are found at the tips of the bacterial cell. Subterminal spores naman po are found near the ends or tips of the bacterial cell. And central spores naman po are found in the center. Now, the placement of the endospores may be used for the identification of some microbial species. For example, your Clostridium tetani usually produces terminal type of spore, while your Clostridium botulinum naman produces subterminal spore location. Now, example naman po of central spore or bacteria that produces central spore is your Bacillus anthracis. Now, in terms of microscopic identification of your spores can be visualized as unstained areas po within the cell using the routine gram staining technique. However, it can also be visualized under the microscope using special spore stain techniques such as Schaefer-Fulton staining technique. So, in the microscopic analysis of your spores, it is a highly refractile bodies in the cell, and then the most commonly used endospore stain is Schaefer-Fulton. So making the spores po usually in the Schaefer-Fulton appears as color green, no? And then the bacteria po is color red. Now, as you can see here, no, uh, it shows an example of the terminal spore of your Clostridium tetani. Okay? Giving the bacteria or the organism a characteristic tennis racket shape or lollipop shaped appearance. Now, we are now done in discussing the different cytoplasmic structures of your bacteria. Let's now go to the cell envelope, which comprises of the outer and inner membranes or outer and plasma membrane, cell wall, and also the periplasm. Please take note that the outer membrane and the periplasm is only present in gram-negative bacteria. Okay, now the plasma membrane or the cell membrane is composed of phospholipids primarily in proteins, but usually they do not contain the lipid sterol. Okay, so unlike the plasma membrane of eukaryotic cells. Now, in exception or an exception po to this is the mycoplasma, which contains sterols po in the plasma membrane. Now, it is also in this structure in which energy is generated, as it is the site of the electron transport chain in your bacteria. Okay. So the site of respiration and photosynthesis is also in the plasma membrane. So it acts as also as an osmotic barrier. So prokaryotes have a high osmotic pressure po kasi inside the cell. So it also acts as the osmotic barrier. So again, the plasma membrane or cell membrane of your bacteria functions as the mitochondria, the Golgi complexes, and the lysosomes of your bacteria. The next structure naman po is the cell wall, so which maintains the shape of the bacterial cell. Now, commonly, the cell wall is classified according to their staining reaction with the gram staining technique. So, the cell wall or the composition of the cell wall is composed primarily of disaccharide pentapeptide units and teichoic and lipoteichoic acid. So, it contains the peptidoglycan or also called as the peptidoglycan murine layer because it contains peptides or proteins and sugars combined. Okay? So typically the cell wall po, it is the main part of the cell that determines that two major types of bacteria based on the cell wall composition, which is your gram-positive bacteria and the gram-negative bacteria. So to have an overview lang po, gram-positive appears as purple bacteria in the microscopic analysis, and gram-negative naman po appears as pink bacteria. Okay? Now, the mechanism on how these stains stick to the different composition of the cell wall of these two types of bacteria is discussed or will be discussed in the laboratory portion. Okay? So, another species po, your mycobacteria, although it doesn't have the typical cell wall of most of your bacteria, it is considered under the gram-positive grouping. Okay? But your mycobacteria has a special type or modified cell wall containing an acid-fast cell wall also, a type of waxy layer. Okay? Now, another naman po, mycoplasma, it doesn't have a cell wall. Okay? May mga ganun po tayong bacteria that doesn't have cell wall. So typically, the term used for bacteria that do not have cell wall is called atypical bacteria. And one example of that is your mycoplasma together also with your urea plasma as well. Now, the cell wall po of your bacteria no, prevents the lysis of the cell due to osmotic pressure. And it is where the flagella also, a structure used for locomotion or movement, is anchored. Okay? So, also cell wall it determines po, as previously mentioned, the staining characteristic of your bacteria. Now let's now discuss the two variations of your cell wall, no? The gram-positive and gram-negative cell wall. Now the first type of cell wall is the gram-positive cell wall, which appears bluish or purplish after the gram staining procedure. Now this type of cell wall po is characterized by a thick layer of peptidoglycan, which is composed of polysaccharide chains of alternating N-acetyl D-glucosamine and N-acetyl D-muramic acid or the NAG and NAM. Now, antibiotics which prevents the synthesis of peptidoglycan is effective against gram-positive bacteria. May example of this antibiotic is sir penicillin. Okay? So gram-negative bacteria which have thinner layer of peptidoglycan naman po is less affected from these types of antibiotics because additionally it has a protective layer called the outer membrane. Now, to maintain the structure of the peptidoglycan layer, now there are short chains of peptides that are attached to each N-acetyl D-muramic acid. Now, the polysaccharide chains are then cross-linked to form a thick network and aids in the stability of the cell. Okay? Now, please take note that teichoic acid and lipoteichoic acid, which are anchored to the cell and to the plasma membrane respectively, are unique po to the gram-positive bacteria. Now, teichoic acids po or your TA, no, are usually anionic polymers. No, pag sinabing anionic polymers, these are negatively charged, which is found in gram-positive bacterial cell wall and usually are made of polyglycerol phosphate units, and they are involved no, in the regulation of cell morphology as well as in cell division. In terms of stability, your teichoic acids po stabilizes the NAG and NAM po in between them. Now, your lipoteichoic acids naman, however, no, it anchors naman po the whole structure of the cell wall into the cell membrane. So this work led to several hypotheses of LTA function to support bacterial growth, cell division, and separation, as well as ion hemostasis, as well as envelope assembly and integrity. So as you can see in the illustration, teichoic acid anchors po the NAG and NAM molecules, while your lipoteichoic acid naman mainly anchors the whole structures of the cell wall of your gram-positive bacteria into its cell membrane. Now, to have an overview, this is a side-by-side diagram of a gram-negative and gram-positive cell wall. So please notice that in gram-positive microbes, there is a thick layer of peptidoglycan surrounding the plasma membrane. While in the gram-negative cell wall, it contains an additional outer membrane made up of lipids, and in between the two uh, membrane containing lipids, the outer and inner cytoplasmic membrane, there is a thin layer of peptidoglycan. Now, as previously mentioned naman po, na the cell wall of gram-negative microbes have two layers. So, gram-negative cell wall contains an inner peptidoglycan layer but is much thinner po compared to those found in gram-positive bacteria. So, it also contains periplasmic space, so involved in the peptidoglycan synthesis, no? And then, it contains an outside peptidoglycan layer, outside the peptidoglycan layer, however, is in outer membrane, so which contains proteins, phospholipids, and lipopolysaccharides or LPS. Now, the protein channels called porins are also present in the outer cell membrane, allowing for water-soluble molecules to enter the bacterial cell. Now, the outer membrane po of your gram-negative bacteria has the following functions. First is it acts as selective barrier, no, by not allowing your harmful substances to enter the cell, while also allowing some molecules to pass through. So it also helps in the bacteria to cling to or attach to host cell and mediate infection. So it also provides a vital function in evading host defenses because it contains strong negatively charged outer membrane, no, which is important factor in evading phagocytosis naman by your phagocytic cells. It is also considered as the endotoxin. So it also comprises the outer membrane po, the endotoxin molecules. So responsible naman po for the virulence of most of your gram-negative bacteria. Now again, the lipopolysaccharide found in the gram-negative cell wall or cell membrane has three regions. So the first region is the antigenic O, no, specific polysaccharide or the antigen O, the core polysaccharide, and the inner lipid A, which is also called as endotoxin. So lipid A po is responsible for fever experienced by those patients infected with the gram-negative bacteria, as these uh, composed primarily of those substance that produces toxin. So it produces fever and shock conditions in patients, which is your lipid A. Now again naman po, an area naman called periplasmic space is also present in gram-negative bacteria. So it contains proteins and enzymes which is needed naman for bacterial survival. So the inner membrane is composed of a thin peptidoglycan layer. So this is the reason for high susceptibility to mechanical breakage for your gram-negative bacterial cell membrane. So mas madali pong masira yung ating gram-negative cell membrane kumpara po sa mga gram-positive because of a thin peptidoglycan layer. Okay? So it involves again it involves no in peptidoglycan synthesis, your periplasmic space. Now, in this table po, shows you a comparison between gram-positive and gram-negative cell walls. So, in the gram-positive cell walls, again, the peptidoglycan layer is thick and the presence of teichoic and lipoteichoic acid can be noted. Also, note the cross-linking between the polysaccharide chain of the peptidoglycan. Okay? So, in terms of shape naman, your gram-positive bacteria typically appears spherical, pwede rin pong rod shape or pwede rin pong filamentous. In gram-negative bacteria naman po, it can appear in forms of spherical forms, oval, no, straight or curved rods, helical, or filamentous. Now, in terms of metabolism, please take note, your gram-positive bacteria in general usually are chemo-organo heterotrophic. So this term po will be discussed further at the last slides in the portion of bacterial metabolism. However, your gram-negative bacteria is quite diverse in terms of metabolism. So your gram-negative bacteria can do phototropic, chemolithoautotropic, or chemo-organo heterotrophic metabolism. So again, your teichoic acids, your lipoteichoic acid and teichoic acid is only present in gram-positive bacteria because it has a thick peptidoglycan layer. Now, endospores naman po typically are present in some groups of gram-positive bacteria, and there is no gram-negative bacteria that produces endospores. Okay? Periplasmic space naman po is present in gram-negative bacteria to produce the thin peptidoglycan layer. Flagellar structures po usually is present on both types of bacteria. However, in terms of complexity uh, or containing the basal ring, no, or that supports the flagella, so for gram-positive, it only has two basal ring structures. For gram-negative naman po, it has four basal ring structures. Now, in terms of resistance to physical disruption, gram-positive bacteria has the higher resistance po in terms of physical disruption due to its thick peptidoglycan layer. Now, in terms of resistance naman po, however, to lysosome or lysozymes, no, your gram-negative naman po, mas madali po silang mas resistant po sila sa mga lysozyme. Now, in terms of inhibition by basic dye, mas madali pong nai-inhibit yung growth ng ating gram-positive bacteria in terms of exposure to basic dyes or stain. Now, in terms of reproduction, both of your bacteria produces or reproduces through binary fission. Now to add po in the discussion of cell wall, now some of your bacteria also produces another type of cell wall which is found in Mycobacterium and Nocardia species, which are gram-positive cell wall. So sometimes they contain lipid contents now called the mycolic acid. Okay? So since lipids are hydrophobic in nature, this mycolic acid affects the permeability of the cell, making it challenging to stain with routine gram staining techniques. Okay? So that's why these type of cell walls are called acid-fast cell wall. Okay? So more than 60% of the cell wall is lipid containing mycolic acid po. Okay? And again, this renders the bacteria hydrophobic in most of your basic or water-based stain. Okay? Now, how do you visualize this type of bacteria containing this type of cell wall? So, in order to visualize microscopically the cells now belonging to such species like your Mycobacterium and Nocardia, no, this is you use po the acid fast staining technique. Now, this is the reason why these microbes are called acid fast bacteria. Now, in acid fast staining, the bacterial smear is first stained with carbolfusin, a type of stain, the colorizer called acid alcohol, and counterstaining with another stain, now methylene blue. Now, acid-fast microbes appear red usually, and non-acid fast bacteria naman po appears blue. As you can see in the diagram, so typically talaga the cell wall of your acid-fast are gram-positive, but additionally, it contains a mycolic acid layer, which renders the outside layer resistant to the typical gram staining technique. Now, some microbes also do not have cell walls altogether, such as those that belong to the genera Mycoplasma and Ureaplasma. Now, such species do contain sterols po in their plasma membranes. They are much similar to their eukaryotic counterparts. Now, microscopically, they appear in various shapes as it is the cell walls function to give shape and rigidity to the cell. So, wala po kasi silang cell wall to form its shape. So usually they grow as L forms in the media or various shapes po. Now, however, no, so some bacteria with cell wall also can lose their cell wall. So there are different forms of your bacteria with defective cell walls. So first is your protoplast, so which are derived from gram-positive bacteria that loses their cell walls. So kailan po ba nagiging protoplast ang ating mga gram-positive bacteria? Paano po sila nawawalan ng cell wall? So this is done by exposing the bacteria to the enzyme called lysozyme, so which weakens the peptidoglycan layer of this gram-positive bacteria. Now, the gram-positive bacterium assumes a shape of a sphere. Okay? So since the cell wall is lost, the bacterial cell is prone to osmotic lysis or shrinkage depending on the concentration of the surrounding solution. So it requires hypertonic conditions for maintenance of these types of protoplasts, a type of forms or shape or forms of these gram-positive bacteria without their cell wall component. So another form is your spheroplast. So it refers naman to gram-negative bacteria that lose their cell walls. So the major difference is that protoplasts now have a single membrane surrounding them in contrast with spheroplast which has two membranes surrounding them. Okay? Because gram-negative nga po has outer and inner membranes. Okay? So they retain some residual but non-functional cell wall material, and osmotically they are fragile, and they are produced by growing your bacteria or gram-negative bacteria with culture media containing penicillin, a type of antibiotic. So they must be maintained naman in hypertonic medium. Same goes with your previous form. Okay? Your protoplast. Now, and the last form would be the L forms. The L forms naman po, ito po yung mga forms of your cell wall deficient forms of your bacteria naman. So, usually produced in the laboratory. So, sometimes spontaneously, they are formed in the body of patients treated with penicillin. So, but they are more stable than protoplast and spheroplast. And they can replicate in ordinary media. Okay, let's now proceed now to the different cell appendages. When we say appendages po, it's not located na inside or within the cell membrane of your bacteria, but rather, these appendages po can be visualized outside the bacteria natin as protrusions po. Now, first of these cell appendages is your glycocalyx. Now, glycocalyx is the outward complex of polysaccharides on certain types of bacteria, which is usually located at the surface po of your bacteria and other cells also. So, the main function of glycocalyx is to help your bacteria in attaching to the surface of tissues or solid objects. There are two types of glycocalyx. So your glycocalyx can appear as capsule or it can also appear as a slime layer. Now your slime layer are similar to capsule but they are much thinner, less organized, and are diffuse layers surrounding the cell. Your slime layer can also act as virulence factor as they aid in the inhibition of phagocytosis and adherence to the host. Now, to better differentiate capsule and slime layer, let's first discuss specifically your capsule. Now, your capsule is another feature found in some species of your bacteria, and capsules are structures that envelope, no, the cell and aids in mediating infection by preventing phagocytosis of the host immune cells. Now, the capsules do not normally retain color with the use of routine stains, so they appear as clear area of halo surrounding the organism. Now, usually your capsule po, to differentiate it from slime layer, it is a discrete organized polysaccharide covering your bacteria to help your bacteria firmly attach to the cell, which is firmly attached to the cell wall. Okay? So typically po in order to to detect no cell wall antigens using serologic test, the capsules po are normally removed from the suspension of microorganisms by boiling process. Okay? So this does not ordinarily stain again with the use of common laboratory stains such as gram or India ink. So may mga special stains po tayo that is used to characterizer capsule po. One example is your his method of capsule staining. So, ang itsura po ng capsule natin sa his method is that it appears as a clear halo surrounding the stained organism. So as previously mentioned po, your slime layer naman is just like capsule pero mas ano po siya, loosely attached to the cell wall. Okay? And it is much thinner. Pareho lang din po sila ng capsule as it is also made out of polysaccharide and it also serves to inhibit phagocytosis and it also helps your bacteria to adhere to the host tissue or to synthetic implants. Okay, another cell appendage is your flagella or flagellum. Now, your flagella po, no, sometimes your bacteria contains this one, no, for locomotion and survivability. So this is the organ of locomotion and survivability. And in terms of structure, no, flagella are whip-like structures po that aid in locomotion. Now, the number and location of flagella within the bacterial cell can vary within different species. So your flagella po in exterior contains protein filaments that rotate and cause bacteria to be motile. So bacterial species vary in their possession of flagella from none as non-motile. And kapag ang bacteria natin is merong flagella, it is termed as motile bacteria. Okay? Now the flagella extends from one end of the bacterium are polar, called polar. Okay, occur singly at one or both ends or multiply in tufts at one. Now, naturally, your flagella po is usually attached to the cell wall of your bacteria. Okay, and typically it is stabilized by a basal body. Okay, and the number of basal body is usually um, used no to identify your gram-positive or gram-negative bacteria. So typically, gram-positive bacteria that contains flagella contains basal rings, while your gram-negative naman po contains four basal rings. Okay, some bacteria also has a unique characteristic of motility which aids in the identification of that bacteria. For example, cyanobacteria, myxobacteria, and capnocytophaga has a characteristic gliding motility. Okay, so the first method that is used to identify whether your bacteria has flagella or not is using the hanging drop method. Okay? So developed by a scientist. Okay? So also no some bacteria also is attracted no in certain stimulus which is called a taxis for this type of motility. So as you can see here in the slide no some bacteria is detected with for its motility by a twitching movement. Also, class, no, we can also use the number and placing of your flagella to identify certain types of bacteria. So some bacterial flagella are called atricus, no, kapag po yung bacteria natin doesn't have flagella at all. So those with a single flagellum naman po at one end are called monotricous bacteria. Okay? So when a group or tuft of flagella naman po is present at one pole of the bacterial cell, it is said to be Lophotricus flagella naman. So when a flagellum is present in both ends naman po or poles, it is classified as Amphitricus. Okay? So when flagella naman sprouts from all sides of the bacterial cell, it is classified as Peritricus. So most of your gram-negative bacilli na motile contains Peritricus flagella. Now, lastly, when tufts of flagella naman po are present on both ends of the bacterial cell, it is called naman po Ampylophotricus. Now, the last cell appendage naman po is your pili or fimbriae. Now, your pili po are hollow structures that can be found on the surface of cells. So there are two types, no, the common and sex or conjugation pilus. So these appendages are not for motility, no, and they are not used for motility po ng ating bacteria. Okay? So usually, no, that is used to connect two bacterial cells and mediate DNA exchange. Also, it can also be used by your bacteria to attach to a certain surface or cells. Okay, so in terms of type again, no, the common or somatic pili is used as a virulence factor of your bacteria or certain type of bacteria, and it is also used for as an organ of attachment. Examples of bacteria that produces common or somatic pili is your Neisseria gonorrhoeae and Pseudomonas species. Another type of pili or fimbriae is your sex or conjugation pilus, which is commonly used naman po for to transfer genetic material or through the conjugation process. Now, this picture shows you the difference between a pili and a flagella. Please take note that flagella is much longer, no, compared to your pili or fimbriae. Now, in this slide po, I have provided you a table summarizing certain commonly encountered motile and non-motile bacteria. So, please take note, some examples of motile bacteria are those bacteria under your Enterobacteriaceae family, no, like your Escherichia coli, no, Pseudomonas species, and some others. Okay? So, those that are non-motile bacteria naman po are your Klebsiella pneumoniae, okay? Your Bacillus anthracis, and your Staphylococcus and Streptococcus species. So, one fact, no, that you need to note is that please take note that if your bacteria is capsulated, it doesn't have the capability to produce a flagella. So, expect nyo po na kapag ang isang bacteria is capsulated or it can produce capsule, so it cannot produce naman po flagella. For example, Klebsiella pneumoniae is a type of bacteria that is capsulated and it is known as a non-motile bacteria. Now, the commonly encountered naman po na capsulated bacteria are your Klebsiella pneumoniae, Bacillus anthracis, and Streptococcus pneumoniae. So, as I have mentioned earlier, no, so those that are capsulated bacteria doesn't have the capability also to produce flagella. Hence, they are considered as non-motile bacteria. So, examples naman po ng mga bacteria that can produce spores, okay, are your Bacillus and Clostridium species. Now, also, I have also listed here those bacteria that produces specific inclusion bodies or achromatic granules. Like, for example, your Corynebacterium, no? Mycobacterium, your Nocardia and Actinomycetes, and also your Pasteurella and Bordetella. Okay, let us now discuss the part wherein we talk about the simple bacterial morphology in terms of reporting also and its microscopic morphology. Now, since there are a lot of different types of bacteria class, we use different classification schemes to identify them and place them into taxonomic groups. Now, listed are the following criteria for bacterial classification. So, some uses the appearance of the microbe microscopically and macroscopically, such as cell morphology, its staining characteristic, cultural characteristics po. Okay? So, others rely on the biochemical test to see differences in the biochemical reactions, and others utilizes antigen antibody reactions for identification. And lastly, no, some uses genetic sequencing to gather information. Okay, let's now discuss the commonly encountered shapes of your bacteria microscopically. Okay, so please take note that your bacteria varies in size, no, from 0.4 uh, to 2 micrometers. Okay, so different bacteria exhibits different shapes depending on the species. So some appear as spherical, which is we note your bacteria if it is spherical as cocci bacteria. Okay? And then some are elongated to rod shapes, and these are called bacilli naman. So those that exhibit spiral patterns naman po are called spirochetes. Okay? So this figure in this slide shows you the different arrangement also of this basic shapes of your bacteria. So your cocci can be called as clusters if you see that the individual cells of your cocci bacteria, no, grows together, no, and form clusters of arrangement. You can also call your cocci as in chains if you see that the cells po growing together are in the form of chains. Okay? So some cocci also can grow in pairs, no? This is also called as diploid or diplococcal cells, and some others form in pairs of four, no, or in tetrads. Your bacilli naman can be called cocobacilli if it is a if the size po is smaller, no, compared to a normal bacilli. You can also call your bacilli as fusiform or palisading depending on a specific arrangement. Now, your cocci bacteria may be seen under the microscope in different arrangements again. So, depending on the species. So, they may appear as singly, no? Pag sinabing singly po, a majority on your bacterial sphere, no, you can observe single growth of your cocci cells. Okay? So, in pairs naman po, kapag tigda dalawa po sila. In chains, kapag nakita nyo po, they are in line in one direction, and you can also describe the arrangement of your cocci as clusters. Okay, so these characteristics also aid in microbial identification process. Now, bacilli are rod-shaped bacteria naman po that vary in size and in length. So their ends may be squared, no, rounded, or tapered when seen under the microscope. So when a single pure culture of a microbe was smeared, stained, and viewed microscopically, and the cells appear differently from each other, it is said that the microbe naman po is pleomorphic. Okay, so pag medyo iba-iba, no, yung shape po niya, hindi, wala pong definite po or defined shape, like cocci or bacilli, you can report it as pleomorphic bacteria. Now, as with cocci po, again, your bacilli can be seen in different arrangements. So, they may occur singly, okay, in chains, no, or side by side, also known as the palisading. Okay. Now, spirochetes, on the other hand, are those with spiral shape and also vary in length and number of helical turns. Now, Treponema pallidum, which is the causative agent of syphilis, and also Leptospira interrogans, which is the causative agent of leptospirosis, are those that are common examples of spirochete bacteria. Okay, we are now done discussing the different morphological structures of your bacteria. Let's now proceed now on discussing the microbial nutrition. Now, microorganisms, no, like us, no, require also specific nutrients which are essential for their growth and multiplication, just like our cells. Okay? Now, the elements such as oxygen, Carbon, hydrogen, nitrogen, as well as sulfur are some of the needed substances in energy production and biosynthesis. Moreover, temperature, pH, and also moisture are also necessary to facilitate their growth and survival. Now, what are the important environmental factors or physiologic requirements of bacteria which influence their growth rate? So, first is your pH, no? This is what the physiologic requirements of bacteria to grow. Another is temperature, and another is gaseous composition of the atmosphere. Now, in the microbiology laboratory, no, we often culture microbes so we can study them. This is why factors that influence their growth must be well understood by the analyst like us. We are the analysts. Now, these important environmental factors, no, must be given attention if we wish to culture your microbes, like your pH, temperature, and the composition or gas composition of the atmosphere. Let us now first discuss the pH. Now, according to pH, no, pH scale is a measure of the hydrogen ion concentration. So we can call certain types of organisms according to their pH tolerance. First is that we call your bacteria or organisms as acidophiles if they can tolerate or grow between the pH 0 and 5.5. Okay? The examples of acidophiles are the following. These are your Picrophilus and Sulfolobodus. Now, another is we can call your bacteria as neutrophiles if they can tolerate or grow between 5.5 naman po to 8 pH. So most clinically significant bacteria that we can encounter in the hospital setting are falls under the neutrophiles. Now we can call naman for your bacteria as alkalophiles if they can grow between 8.5 and 11.5 pH. Examples of these bacteria naman are your Bacillus alkalophilus and your Natronobacterium. So, but also please take that the optimum pH for most pathogenic bacteria is around 6.5 to 7.5. Kaya po yung mga culture media natin is adjusted to a final pH between 7 to 7.5 to well compensate with the optimum pH for your pathogenic bacteria. Now, another physiologic requirement of your bacteria is according to oxygen requirement. So, generally, in terms of oxygen requirement, we can call your bacteria as aerobes or anaerobes. So, to define lang, aerobes po are organisms that require oxygen, no, and grow well with room air. So, your room air po kasi contains about 15 to 21% oxygen and less than 1% carbon dioxide. Examples of aerobes are the following: your Bordetella, Brucella, no, as well as Mycobacteria and Pseudomonas are all considered as aerobes. Now, to define naman po anaerobes, ito naman po yung mga bacteria natin or organisms that do not require oxygen to grow and survive. Okay? But in the presence of carbon dioxide, it can increase the growth of these types of bacteria. Now, there are certain types also of your anaerobes, okay? So, meron po tayong tinatawag na obligate anaerobes, okay? So, these obligate anaerobes po do not require normally the presence of oxygen and they die after prolonged exposure to air. Examples of obligate anaerobes po are your Clostridium and Bacteroides. So, for these bacteria po, um, titignan natin po yung percentage po ng oxygen po sa environment. Dapat po wala po siya. Okay? So pag obligate anaerobes, totally dapat wala pong exposure to any amounts of oxygen. Same goes with your obligate aerobes. Ito naman yung mga bacteria po natin na kailangan po wala pong presence halos ng carbon dioxide. These are your obligate aerobes. Okay? So another type of anaerobes are your facultative anaerobes. Okay? So, ito na po. So, dito naman po nagfo-fall lahat ng clinically significant bacteria natin under the facultative anaerobes. Okay? So, these organisms in definition naman grow either in the presence of or presence, no, of or absence of oxygen. So, hence, they are considered as aerobes that can grow anaerobically. Okay? So they do not require oxygen but grow better in its presence. Example of your facultative anaerobes are your Enterobacteriaceae family. Okay? So obligate aerobes and facultative anaerobes contains usually superoxide dismutase and catalase enzyme, which are protective enzymes that counter toxic effects of oxygen. Okay? Another type of anaerobes are your aerotolerant anaerobes. So in definition naman, these organisms can survive in the presence of oxygen but unable to perform metabolic processes unless situated in an anaerobic environment. So, ito yung mga bacteria naman na um, nag stop po yung kanilang metabolic processes pag tinanggalan mo sila ng pag tinanggalan mo po sila ng oxygen or carbon dioxide. Pero babalik po yung kanilang metabolic processes kapag binalik mo sila sa anaerobic environment. These are Lactobacillus and Cutibacterium. Now another type of oxygen requirement is your microaerophiles. Now microaerophilic bacteria naman po requires decreased oxygen for their growth. Okay? So take for example your Campylobacter jejuni. So that requires 5 to 6% oxygen lang. Your Campylobacter jejuni causes your foodborne infections and diarrhea. So usually po microaerophiles requires in the range of 2 to 10% oxygen lang po for growth. Okay? Okay. Now, one practical technique in the laboratory that we do, no, to identify the gas requirement of your bacteria is through the use of your thioglycolate broth. Okay, usually po yung mga bacteria natin, nilalagay po natin in a culture medium called thioglycolate broth to see their specific growth placing no on this type of media. So, for your obligate aerobes usually, no, they usually grow lang po no on the very top of your glycolate broth, signifying that they need no a lot of oxygen for their growth. Now sometimes also no your facultative anaerobes can be detected in the thioglycolate broth as we observe po no um dispersion no of the growth of your bacteria on the whole thioglycolate broth, but we can see naman po increased amounts of growth on the very top portion of their thioglycolate broth. Obligate anaerobes on the other hand naman po, we can tell that a bacteria is an obligate anaerobe if they are grown or growing lang po on the very bottom of your thioglycolate broth. Now, an aerotolerant anaerobes naman no is detected if the thioglycolate broth is turbid all throughout the setup. Now, microaerophiles naman po lastly is detected if the growth of your bacteria is at the center or at the center of the thioglycolate broth. Now, lastly, based on gas requirement is according to carbon dioxide requirement. So, this one naman po, it matters naman po yung mismong concentration ng carbon dioxide supplied in your bacteria. Okay? So, the term we use usually for those bacteria that require an increased amounts of carbon dioxide, around 5 to 10% of carbon dioxide, is capnophiles. So, capnophiles po are those that require a greater amount of carbon dioxide for growth. Okay? So, most aerobic and facultative aerobic bacteria only needs lang kasi a lesser amount of carbon dioxide. So, examples of capnophilic bacteria natin is your Haemophilus influenzae, Neisseria gonorrhoeae, and Streptococcus pneumoniae. So, one practical setup naman po that we do to facilitate capnophilic environment is using the candle jar technique, which increases nga po carbon dioxide in the environment. So according to nutritional requirement naman po is your as to carbon source po, as to the energy source, and to as to the electron source. Okay? So as to carbon source class, we can call your bacteria as autotrophs or heterotrophs in that sense. So, in definition, autotrophic microorganisms use carbon dioxide as their sole source of carbon. Now, heterotrophs, on the other hand, naman, utilize reduced preform or organic molecules from other bacteria. Now, as to the energy source, we can call your bacteria or microorganisms as phototrophs. No, these phototrophs po are organisms that use light, no, as their energy source. Chemotrophs, on the other hand, makes use of the energy produced by the oxidation of organic or inorganic compounds. Now, lastly, no, as to electron source, we can call your microbes po as lithotrophs, which have the capability to reduce inorganic molecules to be used in the biosynthesis or energy conservation. Now, organotrophs naman po, on the other hand, requires organic substance, no, like carbohydrates and fats, no, for growth and multiplication. Now, to illustrate lang, no, I have provided you this image that shows you how candle jar technique is used to increase carbon dioxide levels for growing capnophiles. So typically, candle jar technique, we literally use white unscented candles, to provide the necessary levels of carbon dioxide for your capnophiles. So usually po, it is done in a makeshift jars, no? And then usually nilalagay po natin dun yung mga bacteria natin for to grow, no, in a capnophilic environment. And then literally we use a lidded unscented white candles, no? Ini-insert lang natin sa loob ng mismong jar. And then we allow the natural extinction of the lidded candle, as the carbon dioxide increases inside. Okay? So notes to remember lang po. So please take note that autotrophs are also considered as lithotrophs because they obtain energy either photosynthetically or oxidatively. Heterotrophs naman po are also called as organotrophs since they obtain energy through oxidation or fermentation of organic substances such as glucose. And tandaan nyo po, all bacteria that inhabit the human body, no, are classified into the heterotrophic or organotrophic groups. And when we say fastidious, no, again, fastidious bacteria needs additional substances such as vitamins, purine or pyrimidines, and hemoglobin for growth and survival. When we say saprophytic naman po, or a saprophytic organism, they require naman po dead organic substances. When we say parasites naman po, they necessitate organic substances from living tissues. Okay, let's now proceed now on the physiologic requirement of bacteria as to according to temperature requirement. Okay? So again, temperature also influences the growth of your bacteria. In fact po, microbes can be classified according to their optimal temperature for growth. Okay? So the optimum temperature for most clinically significant bacteria is 35 to 37 degrees Celsius. Okay? So those that thrives naman po at temperature between 10 to 20 degrees Celsius are called psychrophiles or cryophiles. Okay? So, examples of psychrophiles or cryophiles are your Listeria monocytogenes and Yersinia enterocolytica. On the other hand, okay, or on the other end of the spectrum are the thermophiles naman. Okay? So, which grows best naman at higher temperatures ranging from between 50 to 60 degrees Celsius. Okay? So, those that grow naman po between 20 to 45 degrees Celsius are called mesophiles. And please take note, most bacteria that grow in the human body are called mesophiles. Okay? They have well adapted to the human body temperature and grow best at around 37 degrees Celsius. Okay? So ito po yung basis natin why we set your incubators, no, at around 35 to 37 degrees Celsius. Okay? Those that are called extremophiles naman po are those prokaryotes that able to survive in unusual conditions like the absence of oxygen, increased temperature, and living below the Earth's surface. Okay? So, we use usually the terms thermal death time and thermal death point in terms of temperature requirement. When we say thermal death point, class, this is the lowest or minimum time required naman to kill organisms under a constant temperature. While thermal death point naman is the lowest temperature required to kill organisms in a constant time. Now, this slide lang po shows you the other physiologic requirements of your bacteria to grow. So, syempre, your bacteria also needs moisture. So, according to moisture, No, moisture is vital for bacterial growth and susceptibility testing also. Now, according to salt concentration also, some microorganism can tolerate high amounts of salt, no? So normally kasi hindi po. So pag ang bacteria natin, it can tolerate high amounts of salt, they are called as halophiles. So they require and grow in increased concentration of sodium chloride. Example bacteria that are halophiles are your Staphylococcus aureus and Listeria monocytogenes. Now, barophiles naman po yung ginagamit natin na term if the organism po can grow rapidly in high pressure environments around 600 to 1,100 atmospheres. So, Photobacterium, Shewanella, and Colwellia are examples of barophiles. Okay? So again, no, if your bacteria needs growth factors, or special nutrients, they are termed as fastidious. Okay. So examples of growth factors needed usually by your fastidious bacteria are amino acids, no, purines, pyrimidines, and vitamins. Okay, this would be the second to the last portion of this discussion. Now, in this portion of the lecture naman, we will be focusing on bacterial growth. Now, normally po, again, your bacteria po reproduce asexually, okay, through a process called binary fission. Now, binary fission is different from mitosis class in that the main purpose of binary fission is for asexual reproduction of prokaryotes, while mitosis naman is needed for the growth of eukaryotic organism. Now, however, both processes produce two daughter cells. Now, the generation time, first time it takes for a bacteria to divide, no, into two cells. Now, generation time varies from species to species, as some are fast growers, such as your E. coli, which can take around 20 minutes, okay, for it to divide into two daughter cell. And some naman po are slow growers, like for example, Mycobacterium tuberculosis, that requires a longer period of generation time to produce daughter cells. So typically, a fast-growing bacterium such as E. coli uh, can produce a full growth, no, in a colony morphology at around 16 to 18 hours of generation time. Now, the phases of bacterial growth naman po in a culture media can be visualized po with the bacterial growth curve. Now, in the curve po, we can see that when the environment is rich with nutrients and no toxic products are present, there will be an increase in the number of viable organism. Okay? So, in the growth curve po, ayan po yung phases ng ating bacteria po. Okay? Now, the growth curve po is divided into four phases. So these four phases are the following. So first is the lag phase, also called as the period of rejuvenescence. It is a period po in which there is no cell division and the microbes are adjusting to their new environment, which is the culture media. So once the bacteria adjusted with the environment, it can now go to the second phase, which is the log phase. Okay? Now, in the log or exponential phase class, microorganisms are actively growing and dividing as cell division is most active during this period. So, ito yung literally na stage po na kinakain na nila po yung mga nutrients na nasa culture media. Okay? So, it is also in this period in which the bacteria are utilized for biochemical testing. So, mas better po kasi na dito sa phase natin, sa phase ng bacteria po na log phase, tayo nagte-test. Okay? So kasi ito yung active po na stage ng ating bacteria. Okay? Now, in the third stage naman po is the stationary phase. So in the stationary or plateau phase, there is a balance between the cell division and cell death. So making the number of viable organisms constant. Kaya siya tinatawag na plateau phase. Okay? So the metabolic activities of the cells are slowing down as the nutrients are getting limited. Okay? So, tandaan nyo, nauubos din po yung nutrients na nasa culture media natin. And then, the last is called the death phase or the decline phase, which microbial growth stops as the number of dead cells exceeds the number of viable cells. Then, sa stage na rin na to, no, the death phase, completely, no, sobrang scarce na po ng nutrients po in the culture media. Now, this is the diagram showing the bacterial growth curve. So, the y-axis denotes the number of cells. The x-axis naman is the time. Okay? So tandaan nyo po, the lag phase, no, is the period of rejuvenescence. And then once the bacteria eats the nutrients in the starts to eat the nutrients in the culture media, it will now undergo the logarithmic phase. And then habang kinakain niya po yung nutrients, syempre, so meron na pong balance of dead and viable cells on the stationary. Now, once the nutrients completely deplete and the dead cells increases, it will now undergo the death phase. So, saan po tayo dapat nagtetest, no? Tandaan niyo po on the logarithmic phase. Now, what are the ways or methods to measure the growth of your bacteria in the laboratory? So there are three commonly used measurements to for the growth of your bacteria in the laboratory. First is by cell counting. Okay, through microscopy, no, we can count them cells. So the higher the counted cell, no, in the microscopy, the higher the growth of bacteria. You can also use electronic particle counters or through colony counting methods. Okay? So we can also measure the growth of your bacteria uh by cell mass, no, by weighing the sample or weighing the bacteria, measuring the nitrogen content, and also through turbidimetric techniques. Okay? Pag mas malabo po yung mixture natin, no, it can tell us na mas madami yung tumubong bacteria doon. Okay? And also, or lastly, we can measure the growth of bacteria by measuring or observing its cell activity through biochemical testing. So this table shows you the other methods for measuring bacterial growth. Again, we can use microscopic count, no, to uh to measure the volume of bacterial suspension, no, by counting the bacterial cells literally or directly, no, in a counting chamber or in your microscopic slide. Okay? Plate count also is used, no, to determine naman po the colony forming units of your bacteria in your sample. Okay? So, usually 30 to 300 colonies po should be counted. So in terms of turbidimetric methods naman or cell through cell mass, usually po it requires 10 to 100 million cells per ml of your bacteria to produce turbidity, okay, or a physical turbidity. So this used naman to prepare standard inocula also for antimicrobial testing. Now, the last portion of our discussion would be bacterial biochemistry and metabolism. Now, your bacteria, much like eukaryotic cells, uses also biochemical reactions directed by enzymes in order to break down compounds and synthesize new bacterial components. And it is also used to produce energy, okay, in the form of ATP. So your bacteria also uses ATP for metabolic processes. However, no, different bacterial species can vary on which substrates they can use, which also affects the products produced. Now, such factors can be used for microbial identification. Now, various biochemical pathways now exist in your bacteria for the purpose of breaking down the substrates. And this particular pathway no use determines also the end products. And these end products also, please take note, can alter the pH of the medium itself. Okay? So using these metabolic differences as phenotypic markers is used in the identification of your bacteria. Now, the following criteria for biochemical markers can be used for the identification. First one is the substrate by the bacteria as the carbon source. Second is the production of specific end products, okay, from various substrates which can be determined po in the laboratory. And lastly, the production of an acidic or alkaline pH in the test medium. Now, biochemical tests for microorganisms will be discussed in detail as we go along. Okay? So what I want you to remember, class, for now are these three common criteria. So these criteria are especially useful in the identification of members of your Enterobacteriaceae family, which includes Escherichia coli, Salmonella, and others. Now, typically, your bacteria breaks down carbohydrates no, in order to produce energy, either through fermentation or respiration process. Now, fermentation is an anaerobic process, meaning po it proceeds without the need for oxygen. And this is why no, it is employed by obligate and facultative anaerobes. However, such process po is not as efficient as respiration when it comes to energy production. Now, another note to remember for your fermentation is that fermentation po of carbohydrates no produce a mixture of end products, and such products po accumulate in the medium, which can then be detected in the laboratory. Okay? So analysis of such products are useful for the detection of anaerobes. Usually po, these mixtures of end products are usually different types of organic acids like lactic acid, butyric acid, also production of ethanol and acetone. So these products po of fermentation can accumulate in the medium. Okay? So use loosely in the diagnostic microbiology po yung fermentation natin to indicate any type of utilization. So in terms of utilization din ng sugars, you can whether the bacteria utilize their sugars fermentatively or oxidatively. Okay, and this is detected usually by physically observing the changes in the color of pH indicators, which is incorporated in your culture media. Now, respiration naman po on the other hand, class, is a more efficient process when it comes to energy production. Okay? So as the name suggests, oxygen is necessary. Okay? Because it is the final electron acceptor for the electron transport chain. So this process is employed by obligate aerobes naman and facultative anaerobes. Now, much like in your eukaryotic cells or the our cells, no, cells of humans, no, the starting material also for bacterial oxidation or fermentation is the carbohydrate glucose. So most of your bacteria utilizes glucose for the source of carbon. So when a given bacterial species is capable of using other sugars as their carbon source, it is first converted to glucose muna and then undergoes one of the three pathways listed. Okay? So, commonly pa rin, they can utilize the Emden-Mayer-Hoff-Parnas pathway for glycolytic pathway. Okay? So, also it can utilize the pentose phosphate pathway, okay, or the hexose monophosphate shunt. But especially for your bacteria, some bacteria also can alternatively use the third one, the Entner-Dudorov pathway. Okay? So all convert glucose to pyruvic acid naman. And the pyruvic acid may then either processed with fermentation or oxidation. So if you are asked what are the two types of processing of your pyruvate, so they can be processed fermentatively or oxidatively. Okay, let's now proceed on specifically discussing the third one because the third one is quite special to some of your bacteria. That is the Entler-Dudorov or ED pathway. Now, the Entner-Dudorov pathway was first reported po in prokaryotes by Michael Dudorov and Nathan Entner in the 1950s. So, it is used primarily to catabolize pa rin glucose to pyruvic acid using enzymes naman po distinct either from those used in the on the glycolysis pathway. Now, it can be a major pathway for bacteria growing under aerobic conditions naman. And it is also used as an alternative pathway to glycolysis by some of your bacteria. Now, specifically, your ED pathway uses two specific enzymes, your 6-phosphogluconate dehydratase and 2-keto-deoxyphosphogluconate aldolase to create your pyruvate naman from glucose. To better understand the overview of your Entner-Dudorov pathway is just literally a combination of two common pathways. So the first half of the ED pathway resembles the oxidative hexose monophosphate shunt or the pentose phosphate pathway, and the second half of your ED pathway resembles naman the second half of the EMP pathway or the glycolytic or glycolysis pathway natin. Okay, this shows you the specific biochemical pathway of your ED pathway. So I will not further discuss this, okay? So but please take note that the first half resembles the um pentose monophosphate shunt or the pentose phosphate pathway, and the second path naman po resembles po most likely the second portion of your typical uh, em, uh, typical po na glycolytic pathway. Okay? Now, in this slide naman po shows you the common bacteria utilizing this pathway, ED pathway. So, usually po, your strict aerobic gram-negative bacteria like your Pseudomonas, okay, as well as your Neisseria, can utilize po your ED pathway. Some facultative anaerobes naman na mga gram-negative bacteria that can also utilize alternatively your ED pathway is your Escherichia coli, no, your Vibrio species, no, as well as your cyanobacteria. Okay? And lastly, an example of a gram-positive bacteria naman can utilize your ED pathway is your Enterococcus fecalis. So another note is that all these bacteria presented here, they lack the enzyme essential for glycolysis such as phosphofructokinase. That's why they use the ED pathway instead. Now, to summarize lang po and to conclude, ED pathway is mainly found in gram-negative prokaryotes. Now, your ED pathway also is present in an organism which provides an alternative pathway to utilize your glucose. Okay? And also, please take note your ED pathway is an inducible process. Okay? In the sense that the TCA cycle intermediates are the preferred carbon source in your bacteria. Now, since the ED route generates NADPH instead of NADH, which is required in biosynthetic pathways, it is especially useful po in situations where NADH is limited. Okay? Also, the process is energetically efficient in certain microbial metabolic situations as it requires fewer enzymatic steps compared to your typical glycolysis. Now, also class, there are different types of fermentation. So it depends naman po whether kung ano po yung produce na product in types of fermentation. So first is your alcoholic fermentation wherein it turns your sugar into ethanol and carbon dioxide. So most of your fungi, algae, protozoa, and some bacteria can produce alcoholic fermentation. Another is homolactic fermentation wherein in this process, your pyruvate is reduced to lactic acid which is used to make yogurt, no, pickles, no, and sauerkraut. So it means that only one acid is produced after fermentation. That is called homolactic fermentation. Okay? And then for heterolactic fermentation naman, it can produce other substances other than lactate, like for example, alcohol, formic acid, carbon dioxide, and acetic acid. Okay? So when your bacteria naman produces mixed acid fermentation, it involves naman the production of ethanol and acids such as your lactic, acetic, succinic, and formic acid. So it utilizes formic hydrogen lyase naman po to convert your formic acid into an equal amount of hydrogen and carbon dioxide. That is your mixed acid fermentation. Okay? Now, some bacteria naman po can undergo butanediol fermentation wherein the pyruvate po is converted into acetone that is further reduced to 2,3-butanediol with NADH. Then small amounts of ethanol and mixed acids are also synthesized in your butanediol fermentation. So yung main product po dito is your acetone converted to 2,3-butanediol. Examples of bacteria that can undergo butanediol fermentation are your Enterobacter, Serratia, Erwinia, and your Bacillus. Okay? Then the last type of fermentation is your butyric acid fermentation, which involves the conversion naman of your pyruvate into butyric acid along with acetic acid, hydrogen, and carbon dioxide. Some bacteria naman po that exhibits this kind of reaction are your Clostridium, Fusobacterium, and Eubacterium natin. Now, to summarize lang po the utilization of carbohydrate and the fermentation of lactose. So again po, no, sa ability of microorganisms, no, to utilize sugars for their biochemical processes are key to their identification. Now, sugar fermentation usually produce acid byproducts. Okay? So which can be detected when the culture media in which the microorganism were growing are incorporated with a pH indicator. Okay? So pH indicators change color depending on the pH of the culture medium. So bacteria generally ferments glucose preferentially over other sugar. So glucose must not be present if the ability to ferment another sugar is being tested. So the ability or inability po to ferment the disaccharide lactose is also crucial class in the identification of the members of your Enterobacteriaceae family. So the members are classified either lactose or non-lactose fermenters. Now, in order to process lactose class, the bacterium must have two enzymes. Okay? So, pwede pong, the main enzyme that processes your lactose fermentatively is your beta galactosidase, which catalyzes the breakdown of lactose, no, lactose glycosidic bonds releasing glucose and galactose. Okay? So, one accessory enzyme also that can be present in certain types of lactose fermenting bacteria is your beta galactoside permease, which enables naman po the transport of lactose from the cell wall into the cytoplasm. Okay? So, yeah. So, some bacteria has both these enzymes. Some bacteria also contains only the main enzyme to process your lactose. And some bacteria naman doesn't have these two enzymes at all. Okay? If the bacteria po contains the both of these enzymes, the main enzyme and the accessory enzyme, your bacteria can be called trapped lactose fermenter. If the bacteria naman po only contains the beta-galactosidase, the main enzyme, you can call naman the bacteria as late or slow lactose fermenter. Okay? And then lastly, you can call the bacteria as non-lactose fermenter if it does not have both of the enzymes. Okay? Okay, so this would be the last slide for our lesson for today. So I hope you learn a lot, no, from the basic structure, genetics, and growth and nutrition of your bacteria. Thank you all for listening.